Hello, welcome back to the Computational Genomics Lab. Today we are going to start digging into our results and looking at what happened in those DESEQ comparisons. So just to orient, we are on the Rinn Lab GitHub. In lessons, we have leveled up to 06 and we are running 02 DESEQ start our analyses. Okay, I'm going to open that in my RStudio as an R Markdown file. These are the packages you need, really mostly just DESEQ and tidyverse, but I load my tidyverses individually. It's up to you if you already have the tidyverse package. These will all be in there. And let's load our previous results from 05 where we had our G2S. We're going to need that, and our counts matrix filtered, etc. And let's quick run DESEQ. We did this exact same thing last time, but sometimes it's just good to see it again. We're going to make this mouse long docs DDS. Last time we just called it a DDS. And we're going to use that DESEQ data set from matrix. And we're going to use counts filtered, our sample sheet, and the model we're going to run is based on time. So we want to find genes that are different on time. Same thing here. We're going to run the DESEQ function on that object we just made. Yikes. Oops, that's because I just had a DESEQ highlighted. Sorry about that. If you have just part of it run, it's going to cause an error. So I'm going to go to the end of the line and run it. And there we go. This is what we did before. So it's going to do its thing and run that. And today we're just going to look at the 0 versus 12 time point and see how that would work. And then in the next lesson, we'll do all the comparisons across time points. But when we're digging into the results and trying to figure out what happened, it's sometimes easier just to use a simple example. So today we will find out the genes that change immediately from 0 to 12 hours upon dox exposure. Okay, cool. So let's look at what we got. This will be the same results names as the other function to see what data DESEQ is thinking about. We don't need this intercept term, but we want this 12 versus 0 time point. And notice that it's time underscore point underscore 12. We can use that information to go grab it out of this DDS object, which is going to be kind of a lot to look at for now. So let's go do that. And we are going to, again, much more detail in here about what's going on. But we use the results function to extract one of these. So we did result names to see what results there were. And results itself is going to go in and grab what data there is for that. So we're going to use the results function. Inside the parentheses, we have what object we're going to do that on, the DDS we just made. And this contrast is just something to get used to with DESEQ, is saying, basically tell me what I'm looking for. And we're looking for time point 12 and 0. So that would be this one here. This will get easier when we start doing lots of samples, but this is always handy to know how to grab an individual time point out. And if you wanted 24 versus 0, you could just leave time point, put this as 24, and that is 0. But for now, we're just going to grab 0 versus 12. We'll call that results 0, res 0 here. And breaking down all these arguments that just happened up there. Okay, so now let's make that. Let's see what res 0 looks like. Oh no, it's still in this big DESEQ dataset format. We're going to fix that now by grabbing it out and making it a data.frame. Okay, so this would be really tricky to figure out if you didn't have somebody to help you with this, but like, how do you go from that crazy object into just a nice data frame of data? And so this is what we're going to do. We're going to grab the time equals 12 and the in res equals 0, which there is only time equals 12 in res 0. Again, we're going to do this again in the next class, but this is just a first step to kind of feel like, how do I get into the data? How do I see it? And what is it going to tell me? Okay, so let's do that. Let's make this data frame of 0 versus 12. And cool, look at what we got here. So this is kept the time point, so it knows that it was the 0 versus 12 time point. We get a base mean, which tells us about how abundant the RNA gene, or this gene, was across the samples, how much it changed, the log 2-fold change between 0 and 12, the standard error of that change, and our favorite things, p-values. So let's see, oh look, we have some really significant p-values here. And we will go and try and find those genes. Okay, So we now have a database or data frame of all the genes that changed and their values. All genes that were in there are also in this table, and we can see that here with these 30,000 observations. And so let's see here. But now we might want to do some analysis with this, and so we're going to want to put these row names back into this table. So let's do that with this row names to column function. Again, right now they're in the black or in the meta, and we're going to move them into the data frame. So we're going to do row names from res0df, and we're going to call the new column we're making gene ID. Okay, run that, and voila, that gene ID is no longer in the meta table, it's inside here. And now this is really hard to read. I don't know what gene this is. I'm not too excited about reading that long number, but we made that handy G2S, and we can merge it in. But first, let's do some more typical data wrangling of what you would do. Is first looking for NAs. NAs mean there's nothing there, that the statistics wouldn't work. And let's see here. So is.na is a function that will tell you how many NAs there are. And we're going to use that function to go into res0 into the p-adjusted column and see how many have an NA. And then we're going to sum that. So that answer is going to come out down in our console because we didn't make an object to save it to. Okay. Oh, wow. 15,151 NAs. Okay, so that's just something to keep track of, because when you have numbers in a table and you have NA, those are no longer numbers. They're sort of character values. Okay. So let's see how many rows there are in our result 0df. We kind of already know it's 30,000 from here, but this is another way to check it. With nrow function, let's just see how many rows there are. Okay, 30,000, just as we thought. And now we can find out what percentage of our genes have an NA in the p-adjusted column, saying whether they're significant or not. And usually if there's an NA, they weren't significant anyways, and we'll deal with those later. But this is just kind of some basic R data table skills. Again, we're not going to make an object here. We're going to run a function sum is NA in that column and divide it by the number of rows in the data frame. So just the same two functions above. So about 50% of our genes have an NA. Okay. So now let's merge our G2S, which has our handy gene names here, these gene symbols, gene name. This is kind of, you'll get used to it, but we're pretty much going to use gene ID and gene name, but in this case we have it called gene symbol. Okay, so to do that we just use the merge function in base R, and what it wants to know is what two things to merge. So we're going to merge res0df and G2S, and the thing they have in common is that long gene ID name, this one here. And because we moved that into the data frame of our results, we can now add this G2S column to it. Okay, so we're going to do that. And then so these are the two objects we're going to merge. And what we're going to merge it on is by gene ID here. And gene ID equals gene ID. This C means like print. So it's saying, if we just did one, we wouldn't have to do this, but we're saying the columns are both called gene ID. In this case, it's called gene ID, and in res0, it's also called gene ID. So let's organize it based on that. Okay, let's merge it, and now our res0 df has gene symbol at the very end of the table. But it might be kind of nice to have it right here, so we can organize the columns to do that. Another way to merge, just out of fun, is left join. We'll do the same thing. But to organize it here, we're going to take our results 0df, and we're going to go use the square brackets to go in there and organize the columns. And so we're going to say, I want column 1 to stay where it is, but then I want column 9 to be next, and then 2 through 8 to be after that. And then that way our gene name should be right next to the gene ID. Okay, nice. So that way we can kind of see these two together. It's fine if it was down over there. It's just a little easier to see. Okay, so now we're going to filter. So the next common step in RNA-seq is you would say, I only want genes that are significant of a p-value less than 0.05. Now there is a p-value column and there's a p-adjusted column. I would use the p-adjusted where it's saying it's adjusting by what the distribution of the p-values looks like, and it's cropping out things that could be artificial. So it's the p-adjusted we're going to go after here. And so we're going to make a filtered res 0df because we're now going to change that object, so we don't want to keep it the same name. We're going to put res 0df into this pipe, which then now R is going to be thinking about, and we're simply going to use this function in base R called filter, and we're going to filter with a p-adjusted less than 0.05, and the reason it knows what that is because we piped it in, so it knows all the information in this table. Okay, let's see what we got, and ooh, wow, so if we just filter on p-value of 0.05 and ask how many rows there are, again, we could have found that right here, but just to have a function to do that n row, we see there's 720 genes that are significant. That's a lot. However, we next want to consider that they changed at least twofold up or down in a given time point, and that's what we're going to do here. Is we're going to call this now filtered res 0df underscore 2. Naming is kind of a wild west. You figure out what works for you, but I just want to make sure it has a different name so I can know that I did something different to this res 0df. And we're going to pipe in the original again, and we're going to filter now on two things, and comma is going to separate them. We're going to do p-adjusted of 0.05, and there's a column in there called log2 fold change here, and we spell it identical, and we don't have to use quotes or anything because we've piped in this res 0df here, so it knows the column names. Okay, so now let's see what happens if we say it has to be significant and it has to change a certain amount, and we do that down to 42. Well, wow, that's the answer to life and the universe and everything, says Douglas Adams, but so far we don't know what those genes are. Let's take a look at who they are and what they might be doing. So we're going to call these 42 genes cool genes, and we're going to do this long looking thing, which is actually quite simple, is we're going to make a data frame that just has the names in them. And you're going to want to do this a lot, and it looks bulky, and but it's pretty handy, so that way we can copy and paste into the web, take it out of RStudio and paste it somewhere else. So cat is going to kind of do like we did in bash. It's going to print them out, and we're going to use paste to sort of make them like text. But really what we're looking at here is filtered res 0 df2 gene symbol, and that is filtered res df2 here, and we're going to get this name here, and then we could put it into gene ontology analyses or other things, and that's what we're going to do real quick. So let's just grab those names is all we're doing here. And so this is going to be an object, this cool gene. So now we can see it's like the values here, and we have, oh, we didn't save it. So that just printed it out to the screen here. And so now we could copy and paste right from here, but if the gene list was long, that might be kind of a burden. So let's do as.dataframe in front of that and say go into the filtered res df gene symbol and make each row a new line in the data frame. So we're just going to have one row in the cool genes 12. And cool genes 12 is just so we have the name 12 so we know what time point we were comparing. Okay, so we now have this, and we can copy and paste this and go in, or open it. To be honest, I would pull this onto my desktop and go into Excel. And I encourage you now to go to this website here and paste in your gene names. I'm going to do this the hard way and paste them from right here. And get these 42 genes, but again, we just made a data frame you could export quite easily. And now, so I can just paste these into a text editor. Okay, so I have my gene list, and I'm going to go to this website here. Okay, and I can now paste those genes in here, and see what type of pathways or things that are common to these genes. So let's look in pathways here. It's taking a second to load. Or ontology, so we can see types of pathways they're involved with. I don't know why this is loading so slow. And we'll do this again downstream in our other analyses. Okay, so that's mostly what I wanted to show for today. As some bonus material, you could find all the genes that change in time by doing 0 versus 12, repeating that same exercise for 0 versus 24, 48, 96, and that's a little exercise down here. And if you're just excited to see some plots, we're going to have a whole level on plotting. But you can see here, we can make a quick plot and see where our genes are significant. We're saying here this blue lines are that log 2-fold change threshold we had, and then here's the p-value. And so what we can already see is there's a couple genes out here that are going down over 40-fold, because this is log 2, so it's 2 to the 20. Or much more than 40-fold, very large fold changes here, and these ones have very significant p-values, but they haven't changed that much. And I think that's really good, and now what we'd want to do is do this as a exercise where we take all the results and make these kind of plots. So that's what we're going to do next time, but for now, you now know how to get your results, and you could do this manually for each one. I highly recommend it just to get familiar with the data forms and those R commands. Do the exercise down below, and then on the next lesson, we'll figure out a way to do that even more efficiently.